Hi everyone, we're going to continue talk about the topic. So beside the reaction we mentioned before, the oxidation of fatty acids containing double bonds requires additional steps, as does the oxidation of fatty acids containing an odd number of carbon atoms. In fact, only two additional enzymes, an isomer and reductase, are needed to degrade unsaturated fatty acids. Consider the oxidation of a palmitoylate. So the process is the same as saturated fatty acid until the presence of a double bond between carbon 3 and carbon 4, which prevents the formation of another double bond between carbon 2 and carbon 3. The subsequent reactions are those of the saturated fatty acid oxidation pathway. Fatty acids having an odd number of carbon atoms are oxidized in the same way as fatty acids having an even number, except that propyl-CoA and acetyl-CoA are produced in the final round of degradation. Propyl-CoA converts into succinyl-CoA before entering the citric acid. The pathway from propyl-CoA to succinyl-CoA entails a rearrangement that requires vitamin B12. Propyl-CoA is carboxylated at the expense of the hydrolysis of a molecule of ATP to yield the D-isomer of methylmalonyl-CoA. This carboxylation reaction is catalyzed by propyl-CoA carboxylase. The D-isomer of methylmalonyl-CoA is racemized to the L-isomer, and the substrate for mutase that converts it into succinyl-CoA by an intramolecular rearrangement. The isomerization is catalyzed by methylmalonyl-CoA mutase. Acetyl acetate is formed from acetyl-CoA in 3 steps. 2 molecules of acetyl-CoA condense to form acetyl-acid-CoA, which is catalyzed by sialase. Acetyl-acid-CoA then reacts with acetyl-CoA and water to give 3-hydroxy-3-methylglutaric-CoA, short for HMG-CoA, and CoA. HMG-CoA is then cleaved to acetyl-CoA and acetyl acid. D3-hydroxybutyrate is formed by the reduction of acetyl acetate in the mitochondrial matrix by D3-hydroxybutyrate dehydrogenase. Acetyl acetate also undergo a slow, spontaneous, decarboxylation to acetone. The major site of the production of acetyl acetate and 3-hydroxybutyrate is the liver. These molecules diffuse from the liver mitochondria into the blood and are transported to other tissues such as heart and kidney. During fasting or in untreated diabetics, the liver converts fatty acid into ketone bodies, which are a fuel source for a number of tissues. Ketone body production is especially important during starvation, when ketone bodies are the predominant fuel. High blood levels of ketone bodies result in certain pathological conditions. The most common of these conditions is diabetic ketosis in patients with insulin-dependent diabetes mellitus. These patients are unable to produce insulin. The presence of insulin has two major biochemical consequences. First, the liver cannot absorb glucose and consequently cannot provide oxalic acid to process fatty acid-derived acetyl-CoA. Second, adipose cells continue to release fatty acids into the bloodstream. These fatty acids are then taken up by the liver and converted into ketone bodies. The liver thus produces large amounts of ketone bodies, which are moderately strong acids. The result is severe acidosis. So the decrease in pH impairs tissue function, most importantly in the central nervous system. The principle that synthetic and degradative pathways are almost always distinct. Some important differences between the pathways are as follows. First, synthesis takes place in the cytoplasm, while degradation takes place primarily in the mitochondrial matrix. Second, intermediates in synthesis are covalently linked to the sulfur groups of an acid carrier protein, short for ACP, rather than the sulfur group of coenzyme A in degradation. Third, the enzymes of synthesis in higher organisms are joined in a single polypeptide chain. In contrast, the degradative enzymes are not linked covalently. Fifth, the reductant in fatty acid synthesis is NADPH, whereas the oxidants in fatty acid degradation are NAD+ and FAD. And the last, so the isomer form of the hydroxy acid intermediate in degradation is L form, while the D form is used in synthesis. Fatty acid synthesis starts with the carboxylation of acetyl-CoA to malonyl-CoA. This reversible reaction is the committed step in fatty acid synthesis. The synthesis of malonyl-CoA is catalyzed by the cytoplasmic enzyme acetyl-CoA carboxylase 1, which contains a biotin prosthetic group. Carboxybiotin intermediate is formed at the expense of hydrolysis of a molecule of ATP. The activated CO2 group in this intermediate is then transferred to acetyl-CoA to form malonyl-CoA. The enzyme system that catalyzes the synthesis of saturated long-chain fatty acids from acetyl-CoA, alanyl-CoA, and NADPH is called fatty acid synthesis. The synthesis is actually a complex of distinct enzymes. The elongation phase of fatty acid synthesis starts with the formation of acetyl ACP and malonyl ACP. Acetyl transferases and malonyl transferases catalyze these reactions. In the condensation reaction, a 4-carbon unit is formed from 2 carbon unit and a 3 carbon unit, and CO2 is released. The next 3 steps in fatty acid synthesis reduce the ketone group at carbon 3 to a methionine group. These last 3 reactions, a reduction, a dehydration, and a second reduction, convert acetyl-CoA ACP into butyrate ACP, which completed the first elongation cycle. The sequence of reactions is repeated until the final product, pimetate, is formed. The major product of the fatty acid synthesis is palmitate. The elongation and acetylation of fatty acids are accomplished by accessory enzyme systems. Endoplasmic reticulum system can introduce double bonds into long-chain acetyl-CoAs. For example, in the conversion of like sterocyl-CoA into oleocyl-CoA, a cis double bond is inserted by an oxidase that employs molecule oxygen and NADH or NADPH. The reaction is catalyzed by a complex of 3 membrane bound proteins: NADH cytochrome B5 reductase, cytochrome B5, and sterocyl-CoA desaturase. Fatty acid synthesis and degradation are reciprocally regulated. Acetyl-CoA carboxylase, an essential control site, is phosphorylated and inactivated by AMP-activated kinase. Acetyl-CoA carboxylase is switched off by phosphorylation and activated by dephosphorylation. AMP-activated protein kinase, short for AMPK, converts the carboxylase into an inactive form by modifying 3 serine residues. AMPK is essentially a fuel gauge, so it is activated by AMP and inhibited by ATP. The carboxylase is also allosterically stimulated by citrate. The level of citrate is high when both acetyl-CoA and ATP are bonded, signifying that raw materials and energy are available for fatty acid synthesis. So citrate acts in an unusual manner on inactive acetyl-CoA carboxylase, which exists as isolated inactive dimers. And the citrate facilitates the polymerization of the inactive dimers into active filaments. Okay, so that's all for today, and thank you for your listening. Bye-bye.